All right, everyone, buckle up because today we're taking a deep dive into some really cutting-edge stuff, predicting brain tumors. And you know what they say? Early detection is everything, right?
It really is.
Well, this research we're about to unpack might just hold the key to predicting brain metastases, specifically in lung cancer patients, particularly those with lung adenocarcinoma.
Yeah, this is a big one. What's so fascinating here is how they're doing it. We're talking about decoding the secrets hidden within our DNA.
Okay, now you're just using big words to make me nervous. DNA. Secrets.
It's all about methylation patterns, actually.
Methylation, DNA, decode for those of us who, don't walk around speaking fluent science. Break this down for us. What are we talking about here?
Okay. Picture this. Your DNA is like this massive cookbook
Oh, okay, I'm with you.
filled with recipes for your cells.
Yeah.
Methylation? Think of it like attaching little sticky notes to certain recipes. Some notes might say, like, make this now, lots of it. Others might say, ignore this one for now. And these notes, they control which genes are turned on or off. It's all about influencing how our cells actually behave.
So it's like we have a secret code within our code.
Exactly.
But how does this relate to lung cancer and brain tumors?
That's where this new study comes in, just dropped in Nature Medicine, by the way.
Hot off the presses.
They took a look at these specific methylation patterns. But here's the kicker. They looked in the tumor cells of patients with lung adenocarcinoma. And get this, they found they could predict, with a really surprising level of accuracy, whether that tumor would spread to the brain.
No way.
And not just that,
Yeah.
within 5 years. And get this, it's even more accurate than the traditional staging methods.
Hold on, back up. So by looking at these sticky notes on the DNA, they can tell if the cancer is more likely to spread to the brain.
That's what they're saying.
That's incredible. But okay, how does this actually help patients? What's the real world impact here?
Okay, so let's say you're a doctor,
Okay.
right? You're treating a patient. Early stage lung adenocarcinoma. Okay. Now, typically, you might not be as aggressive with treatment, right? Right. Because the risk of brain metastasis is...
Whoa.
Right. But what if this methylation test, because you can actually test for this, what if it shows a high-risk score?
Game changer.
Right. Treatment plan is totally different now, right?
Yeah, you might consider other options, more aggressive options up front, right? To try and get ahead of it. Prevent those metastases from even having a chance to develop.
Exactly. And on the flip side, let's say someone has a more advanced tumor,
Okay.
but their methylation score is low.
Interesting.
They might be spared unnecessary interventions.
And all the side effects that go with that.
Exactly. We're talking about potentially tailoring treatment plans based on a patient's individual risk. And it's all thanks to these methylation patterns.
That's personalization on a whole new level. But it sounds incredibly complex. How do doctors even begin to make sense of all this methylation data?
Well, the researchers, they developed this tool. It's called a nomogram.
A nomogram, okay?
Basically, it takes this methylation predictor, right?
Okay.
And it combines it with other clinical factors, tumor size, lymph node involvement, that kind of thing.
So it's not just about the methylation.
Right. It creates a personalized risk score for each patient.
So like a percentage chance of developing brain metastases.
Within 5 years.
So not just a yes or no answer, but an actual number. That's powerful, both for doctors and for patients.
Absolutely. Shared decision-making, informed consent, it opens up so many doors.
Wow.
And the best part is, this isn't just theoretical. They actually put it to the test.
Oh, really? So they use this with real patients?
Real patients, yeah. And the results, eye-opening.
You mean like those case studies they talked about in the paper, where they kind of went head-to-head with the methylation predictor and the traditional way of assessing risk?
Exactly. They had this one patient, early-stage lung adenocarcinoma.
Okay.
Something you'd normally look at and think low risk,
Right.
based on the staging at least. But their methylation score, through the roof.
Wow.
And sadly, they did end up developing a brain metastasis.
See, that's what gets me. It makes you wonder how many other people are out there, right? Early stage, being told they're low risk, but maybe this test?
Could be a whole different story.
A whole different story, exactly.
And that's just one example. They had another patient, stage 3 lung adenocarcinoma.
Okay, so more advanced.
Much more advanced. High risk, you'd think, right?
Right, just based on the staging.
Exactly. But get this, methylation score,
Right.
low.
Wow.
And they were brain metastasis free for, get this, over 8 years.
8 years? Wow.
Yeah.
That's That's incredible because we're not just talking about adding more information here. It's like potentially changing the whole approach. The
Absolutely.
peace of mind that this could give people,
Oh,
or
I can't even imagine.
right, both patients and their families. All right, so remember how we talked about methylation being like those sticky notes on a cookbook?
Right, right.
So it turns out there's more to the story.
Oh, there always is.
They were also looking at something called copy number alterations.
Ah, yes.
What in the world is that?
So we've got our cookbook, right?
Okay.
Copy number alterations. That's like someone coming in and making photocopies of entire pages,
Okay.
or maybe ripping some pages out completely. We're talking about duplicating or deleting entire genes.
So not just like subtle changes, but like massive rewrites
Massive rewrites.
of the instructions, essentially.
Yeah. And they were looking to see if these alterations had any connection to brain metastases.
Did they? I'm like on the edge of my seat.
It did. They found that a bunch of these copy number alterations, especially in genes like EGFR, MCL1, and MYC, they were linked to a higher risk.
Okay, so those are the ones to watch out for.
Right. But here's the thing. They also found that deletions in certain genes, like ACTG2 and BLM, for example,
Okay.
associated with a lower risk.
So it's like a two-way street. Some changes increase the risk, some decrease it. It's like this puzzle, right?
It really is.
And we're trying to figure out which genetic variations are sending those tumor cells on a beeline for the brain. But it's not just about the tumor cells themselves, is it? We talked about this earlier, the tumor microenvironment.
Right, right. The tumor's neighborhood.
Exactly.
That plays a role too. And this is where it gets really interesting.
Right, the tumor's neighborhood, what they find out about that.
So remember how we were talking about those immune cell populations?
Yeah, different types of immune cells hanging out around the tumor.
Right. Well, they noticed something interesting about the tumors that spread to the brain. They tended to have more of these cells called cytotoxic T lymphocytes.
Okay, cytotoxic T lymphocytes, those are the ones that like directly attack and destroy other cells, right?
Got it. Like the body's little assassins. And they had a higher ratio of these compared to another type called monocytes.
So it's like the tumor is building its own army. And the makeup of that army influences whether it goes after the brain.
It's a good way of putting it. And speaking of armies and battles, remember those liquid biopsies we touched on?
Oh, yeah. The idea of analyzing those tiny bits of tumor DNA floating around in the bloodstream, no surgery, no biopsies, just a simple blood draw. It's like futuristic stuff.
Right. Well, they use this technique called CF-MEDIC-SEC. Basically, it zeroes in on those methylation patterns we talked about, but this time it's in the DNA fragments just circulating in the blood.
So they're looking for those sticky notes in the blood.
Pretty much.
Yeah.
And get this, they could tell the difference between brain metastases and other brain tumors, like gliomas,
Wow, just from a blood sample.
just from a blood sample, and they were really accurate.
That's incredible. Think about it. People who might not be well enough for a brain biopsy.
Exactly. It could completely change how we diagnose these tumors. Less invasive, more accessible.
This is seriously groundbreaking. But this was all done at one institution, right? What's next? Do these results hold up in bigger studies? More diverse groups of patients?
You're always thinking like a scientist, which I love. Yes, we need larger, multi-center trials to confirm all of this, to make sure it's reliable for everyone.
It makes you wonder, though. They focused on lung adenocarcinoma, but could these methylation patterns be the key for other cancers that spread to the brain?
Now that is the million-dollar question, isn't it? Imagine. Breast cancer, melanoma, any cancer that tends to go to the brain. Early detection, personalized treatment, the impact could be huge.
It's like we're on the verge of this whole new world, being able to predict and personalize treatment for brain metastases in a way we never could before. So for everyone listening, we want to hear from you. What part of this research really stood out to you? What questions are you left with? Keep those brains buzzing, and we'll be back soon with another deep dive.